We are now recording. So without further ado, I will hand over to René Smulders, who can tell us about the design of the ash part of the 4-tree array. Thank you very much, René.
Yeah, thank you, Richard. And I'll try to keep it brief. And first I have to try to share my screen. So hopefully you see it now in presentation mode. Yes?
That's super. Yeah, thank you very much.
So basically, so the Bee Forest was an EU project which, as one of the goals, had the development of a SNP array for ash, poplar, Pinus pinaster, and Pinus pinea. And when the project was submitted, they actually had a discussion with Thermo Fisher, who now owns the Axiom SNP array platform. And basically, Thermo Fisher proposed a two-step developmental process in which in the first step you would test a large number of candidate SNPs on a relatively small panel of candidate genotypes to see which SNPs would perform best. And then in the second step, select the best SNPs and then put them on a smaller array that would be cheaper to use. And they gave the consortium a good price for the whole developmental for both arrays together. So basically, we'll be talking about the screening array and about the design of the 4-tree array that can be bought commercially, and for which then the ash SNPs are relevant for the people here in this meeting. And so Wur was involved as a partner in the Bee Forest project, partly because we have a group in environmental research working on ash with an ash collection and crosses and tests for ash dieback disease. But we were involved because we, in the group of plant breeding, we have experience with SNP arrays. We made Axiom SNP arrays for roses and chrysanthemums, all polyploid species, and we had some experience before that in Illumina arrays for potato and apple, and even various arrays for apple. So back to ash. Initially, we thought we would have to mine SNPs from a transcript reads or something like that. But in fact, there were already so many sequences available in the ash genome that were used for the ash genome, in the sequences used for the ash genome sequence made by Richard Buggs and his colleagues, that we decided to focus on SNPs that already existed. So you selected a large set of potential SNPs, and then next to a random selection of these SNPs across the genome, we also focused on SNPs near interesting genes that might be associated with ash dieback disease and a few other traits. And that sort of settled down in 2,500 SNPs associated with ash dieback disease, a few with emerald ash borer susceptibility, and a few SNPs in candidate regions linked to incompatibility. And then we did some technical filtering. So you don't want 3 allelic SNPs and some other SNPs with multiple hits on the genome sequence. And that resulted in a set of still over 300,000 SNPs. Then I think this partly overlaps, sorry. So in the end, we had potentially 126,000 SNPs available. And then this was the set for the screening array, SNPs for these candidate genes, SNPs from other associations, and then SNPs randomly taken from the genome to cover it as good as possible. So in theory, we had over 130,000 SNPs available for the array. Sorry, something wrong here. And then when you do a test array, you can get all kinds of typical outcomes. What we're typically looking for is what Thermo Fisher calls the poly high resolution SNPs at the upper left corner, where you have a very good separation between the major and minor alleles, and you can also distinguish homozygotes from heterozygotes being the cluster in the middle. And you want to get rid of the other classes in which you cannot distinguish your genotypes. And we had some additional metrics. So in the end, of these SNPs on the screening array, a bit more than 47% was successful in this poly high resolution class. And that was more than 59,000 SNPs. And in fact, there's not room for 59,000 SNPs on the final array, because 50,000 SNPs divided by 4 species left us only room for a little bit over 13,000 SNPs. So on the 4-3 array, this was the final set of SNPs that were both interesting and had a good quality in the test genotyping. So we had 1,700 SNPs associated with these 3 test traits of interest. We have almost 200 SNPs selected to discriminate Vaccinus excelsior from Vaccinus angustifolia, which was a request by Arnaud Dowkiw, or a French collaborator. And he inserted some angustifolia trees in the initial set, the test set. Then we had almost 10,000 SNPs chosen to cover the Vaccinus excelsior genome as good as possible, based on Richard Buck's version of the genome. And then we had some additional high quality SNPs because we had a little bit space left. So in total, we have 13,407 SNPs that technically should be able that should be technically good SNPs that can that you can genotype well. And then in addition, and so recently we had, not recently, already perhaps 1 or 2 years ago, we also got access to a preliminary version of the new genome sequence, the more contagious genome sequence that is generated by a consortium led by Poland. And mapping the SNPs that we had selected back to that genome, 91% of those SNPs have a unique location in one of the 23 chromosomes. So that will be something like a bit over 11,000 SNPs have a position on that genome. And all those SNPs work technically well and are on the 4-3 array. And I think that's basically the end of my presentation. I'll stop sharing. So this was very brief. The design of the array. Yeah. I'm not sure whether perhaps there are some questions, but otherwise.
Well, thank you for being so brief and to time. That was really, really useful for us all. I think we'll save questions till the discussion, if that's okay, because we've got quite a packed schedule. Yeah, sure. Yeah. So I'll hand straight on to the next speaker, who is Carey Metheringham. Carey, please.
Okay. 1 moment. Can you see that?
Yes.
Oh, now we can't.
How about now? All right. So I'll be quite brief on the introduction of where these SNPs came from the Stocks paper, because I think everyone here has at least some familiarity with the pool GWAS that was done on the field trial data. But the brief version is that the genome-wide association study identified SNPs that were more likely to be associated with ASH dieback. And from those, I took the top set to train a genomic prediction model from which you could predict the susceptibility of Ash dieback in a test set of 150 trees. And this prediction was measured by two factors, which was a correlation of the genomic estimate of breeding value as estimated from the predicted effect of all the SNPs, and the accuracy of assignment, which is how accurately it was able to assign healthy trees to the top half and unhealthy trees to the bottom half. So essentially, how often does it get right guessing whether a tree is healthy or unhealthy? Did it just drop off again?
I'm afraid it did.
One would think I don't work with computers and okay, let's hope it continues to understand PowerPoint. So yeah, in the original genetic prediction, we were able to predict the health of the test trees with an accuracy of 67%, either using the top 10,000 SNPs as selected from the GWAS, or by using a subset of 2,500 of those which had the best prediction. So what I did from there on is we submitted those 2,500 for inclusion on the array. And if I'm getting my numbers correct, only 1,633 made it all the way through to the final array, which were removed for various reasons, which I'm sure René can explain better than I can, but a lot of it was potentially due to linkage, because the original set of 2,500 were not filtered for linkage because they came from a pooled population. So there were cases where there was more than one SNP for a linkage block. However, when you look at that set of 1,633 SNPs, even before you start to try and predict any breeding values and predict any tree health, you see that they don't contain a lot of the signal that was in the original set of 2,500. So if you're looking at how much of the additive effect size is contained within the ones that are included in the array, it's only sort of 1.6.
I fear that we've actually lost Carey. Can anyone see Carey? I can just see a camera with a line through it.
I can't see her. I've never seen that symbol before on Zoom.
Oh, she's re-emerged in a different place. Carey, can you hear us?
I can hear you. I apologize for this. It was working fine yesterday when I used it for something else, and it's.
Don't worry, just carry on. We can see you again. I'll unpin the place where you are not.
Excellent.
I think it already is.
Can you see my screen still?
Yeah, we can still see the screen. We've seen that throughout.
Okay.
So
as I was saying,
your screen has just gone.
Okay, I'll carry on. So as I was saying, only a small was contained within those SNPs, and I think that's likely because a lot of the most predictive SNPs
Harry, shall I share your slides? Because you emailed them to me. How about I share the slides? Would that work better?
I think we lost her. Maybe you just show the slide.
I'm going to try without video in the hope that that's going to mean it blinks off less often. I have no idea why it's doing this. I apologize. But I think the reason.
Harry,
Yeah.
I've got your slides, so shall I share them?
Yes, I think that means there's less that can go wrong. I think my computer currently will do Zoom or PowerPoint, but can't cope with the concept of both at the same time, which is quite unfortunate. So I'll have to just go for next slide, please.
Well, let's see if I can get it to work. Sorry, I'm not.
Wow. I was saying before I was rudely interrupted by the computer that the reason that only a small proportion of the predictive effect sizes in those top SNPs is likely because of linkage. If you've got one block of very predictive SNPs where you've only taken one without any retraining, it looks on paper like you're not getting much prediction out of those sites. And what I would like to show you now is the next slide, which contains a box plot. So if you could imagine for yourselves a box plot that shows two boxes that are not very significantly different from each other. And this is when you try and predict the breeding value of those original 150 test trees without doing any retraining on the effect sizes. So just using the original effect sizes and the 1,633 SNPs that were included in the array. And we have slides again. And if you just do the prediction without any retraining, there is no significant difference between the predicted health of the healthy trees and the unhealthy trees. And there we go. And your accuracy of assignment is not really much above 50. And a 50% accuracy of assignment is essentially saying you could guess whether a tree is healthy or not. I'll try and be brief because we've lost some time to technical issues, but if we go to the next slide, please. It shows if you retrain those effect sizes, so recalculate them, retrain the model just using that smaller set, you are then able to get a higher accuracy assignment than without retraining, because you can get up to 54% accuracy, which is still not as good as the original set by a long way, and not that much better than 50-50. I think here we're recovering anything that was lost due to linkage, because now you're piling up all that signal that was once spread out over multiple SNPs onto the one SNP that represents that block. It's still not a particularly great prediction. And the next thing I did was to look at if you include the additional SNPs that were included in the array, which hadn't been used in the original training, and adding those into the model. And this does not improve the accuracy of prediction. This, in fact, makes the accuracy of prediction worse. You're less able to assign tree health, and it's a much broader spread of predicted breeding values. And which is interesting, because usually if you add more SNPs, you get a better value. But in conclusion, I think we've lost a good proportion of that power of prediction with the removal of SNPs, likely due to biological reasons that they can't be included on the array. Many of these are not near genes, they're potentially in repeated regions or parts of the genome that are hard to probe, or just multi-allelic SNPs that aren't suitable for inclusion on the array. And adding in sites that are not previously strongly associated with dieback susceptibility can actually reduce the accuracy, because you're increasing, every site prediction comes with a large amount of error, and here you're increasing the amount of error without actually increasing the amount of prediction, if that makes sense. I'll save questions for the end.
Thanks, Carey, and sorry it took me so long to get your slides up, but well recovered.
Sorry for my issues.
Thanks very much. As you say, we'll save questions and we'll move straight on to people who have actually been using the array in real life, not in silico. So Giorgio Tumino, if I've said that correctly, can I hand over to you, please?
Can you hear me well? I will share the screen. Okay. Can you hear me? Yeah.
Yes.
Okay, great. Okay, yes, my name is Giorgio Tumino. You pronounce it well. I'm working at Wageningen University in plant breeding as a quantitative geneticist. So I will present what we, the activities that we have done within the PE Forest project on ASH, and this is mainly collaboration within us and the group of Arnaud Dowkiw in Rye in France. Just about the recording, I'm not sure whether, so there are also some unpublished results here, so I'm not sure whether you want to put the video into, but just be aware that there are those unpublished results. Okay, so let's see. In this presentation, I will just talk briefly about the genotypes that we have sampled and something about population structure and relatedness we have estimated in our sample sets, little bit on linkage disequilibrium, and then something about the two traits that we used about dieback, which is just the ground defoliation and lesion length after an artificial inoculation, and then the results about genomic prediction and NGVOs. This has been already introduced by René, so I will skip that. What we have genotyped with the 4-tree arrays is there are two sets of totally 2,000 samples. About 900 from Ur and about a little bit more from INRA, and they are including, as said by René, some angustifolia samples and some potential hybrids. So first we did some analysis on population structure. Actually, this work has been done by Sarah Ben Sadoun at INRA. She performed a principal coordinate analysis, and in this picture you can see the first two coordinates, and well, the first coordinate is actually distinguishing between angustifolia samples that are those one in black on the left, and all the other samples in blue and red that are assumed to be fractions Excelsior. But along the X, the coordinate, there are some samples here that are potential hybrids. And when we do the same within Excelsior only, we can see that there's again a principal coordinate analysis that the clustering of samples is pretty well according to provenance of samples, so that's nice to see. We also calculated some close relatedness among the samples, and this has been done by estimating IBD Cotterman coefficients. They are plotted here, K1, K0, and well, just to highlight that this cluster here is a cluster of full SIBs, and we can distinguish quite well them from all the rest of relationships. So it can be used well to do this kind of analysis as well. Very briefly on linkage decay, we have estimated the LD decay over distance. This was done on the Wurth samples only by using different estimators. Actually, what I wanted to show you is just if you look at the mid curve, that's the fit done by the Hillenweir model, and the LD in this case was estimated to decay below 0.2 in 5 and 5 kilobases, so very, very short. And using other methods, let's say the maximum that we have estimated is 26 kilobases, so it was still very, very short. So the lead decay is very fast, between a few kilobases. And if you just do, well, roughly some calculation with this amount of SNPs and 800 megabases, and I'm considering here the largest decay, there are so many, so more than 30,000 blocks, LD blocks over the genome. And with this number of SNPs, yeah, we are just not covering very well them. Well, that's the one point. It's just for maybe later discussion on application on GWAS and genomic prediction. And the two traits that we have used, one is, sorry, is there any? No, this was. So one trait is the foliational crown. I think that more appropriately is called terminal shoot dieback, and they score in these 5 classes. We use this proportion of the foliation from 0 to 1, where 0 is healthy and 1 is a dead tree. And those are the distribution of these traits in the Boer sample at the top and in the ERA sample at the bottom over 3 different years. And you can see also that there are some different, quite a difference between the two sets, because most of the trees in the Boer trial were infected or with, let's say, with many trees with many symptoms, and that was not the case at the IRFA trial. And the other phenotypes that we have used is the lesion length that is measured after artificial inoculation on some clones. And what we measure is that the length of this lesion downwards from the inoculation point. And this was done in 2 different years, but actually there are only 10 clones that were scored in both years. Those scores were very consistent among between the two years, and those are the distributions. In the 2 years, actually, we calculated just the mean over the 2, and we used the whole sample set in the following analysis. And the first analysis was the GWAS on this trait, on lesion length, both using a single locus a model and multi-locus model. Well, overall, it's not successful, I would say. We have just in the multi-locus model, we have these 2 SNPs that were significant. We will investigate that further, perhaps, but they don't seem so robust to me. The same was for the other phenotype, for the defoliation. So we couldn't find any significant association. Now, when we do genomic prediction, this is on the Boer sample as well, using both defoliation and lesion length. And for defoliation, you can see that the predictive ability. This is what has been called correlation in the previous presentation from Carey. So just correlation between predicted values and observed phenotypes, which is very low. While when we use lesion length, we have predictability of around 0.5, which is quite good. And this has been done using these 3 different models, as you can see here. So to conclude, the 4-tree array was quite a powerful tool to study population structure, and the relatedness we have seen was also possible to identify or to distinguish full SIBs from half SIBs. Although there is a very fast decay, that's quite known, but this could have quite a limitation. Let's say could pose quite limitation, especially for GWAS, but maybe also for genomic prediction. And in our case, we have seen that lesion length was quite more predictable than the defoliation. So which is nice. That's, of course, they are not the same trait. So the defoliation is something much more complex, perhaps less accurate, less precise in terms of phenotyping. So that's it. Those were the people who worked in these projects, Sarender now from IRA and all the other colleagues from Buckingham. Thank you.
Thank you very much, Giorgio. That's encouraging. So we'll move to Austria now, to Aglaia Szukala, to tell us about some results from Austria. Thank you.
Hi everyone. Can you see? No. Okay. Can you see my presentation now?
Yeah.
Great. Yeah. So again, thank you very much, Richard, for organizing this meeting. And so I will give you a short overview about the usage of the 4-tree array for our Austrian samples. So Heino Konrad gave recently a presentation at U4Gen about our projects. So I'm not saying too much today, but if you have more questions about it, you can ask afterwards. So the premises for our Ash in Distress project were that we could find healthy trees within highly damaged stands, and actually it was shown that heritability of resistance was quite high. So basically our project aimed to sample hundreds of or select hundreds of resistant trees across full Austria, and we actually selected 715 trees overall that cover quite well the entire distribution of common ash in Austria. Then we raised over more than 35,000 seedlings in our common garden in 4 different sets. These are R1 to R4 that are based on the germination year, and these are fully randomized sets of seedlings. And we actually monitored these seedlings for 3 years for resistance against ash dieback, and eventually we selected 1 healthy and 1 very highly susceptible individual from each original mother trees from this set of 715 trees, or almost all of them. And overall, we sequenced then 1,152 individuals following this setup, and with a 4-tree array, so including 13,407 SNPs. Also showing some results from of genetic structure for our data using the 4-tree array. So basically we could see that 3 main clusters are detected within Austria. Most of the samples fall within such eastern cluster, which contains most of the accessions. There is this western cluster here, and then we also detected some individuals with this high amount of light blue ancestry, as shown in the structure plot, which we hypothesize being probably introgressed from Fraxinus angustifolia, that are mainly found in the most eastern part of the distribution. And yeah, you can see again in the PCA that these are separated somehow from the main cluster, but also this is not a strongly separated cluster. So there are also individuals within this major cluster here that also have this in part this ancestry. But yeah, for most of the analysis that followed, then we actually excluded these individuals as we think are probably introgressed from a closely related species. And I wanted to spend just a few words about our data filtering before we carried out genotype resistance association analysis. So this was our raw data set, as I said, and we filter SNPs according to quality thresholds of the calls, but also we excluded all monomorphic sites and sites with a minor allele frequency below 0.05. And actually these two steps filled out quite a major amount of loci. And overall, we remained with 7,373 SNPs, which actually is a quite strong reduction from the original data set. And what I wanted to note here is that actually I recently noticed that by performing these two filtering steps, we actually removed a majority of those loci that were previously found to be highly associated with the resistance in the Stocks et al. Paper from 2019. And that actually most of these loci, I also checked them visually, several of them, and are either monomorphic in our data or contain very rare minor alleles. So we're basically filtered out already during this step. So they don't seem to be really informative for our ASH in Austria. And I think this was quite an important aspect to notice. So then we went on to perform genotype resistance association. I didn't explain very much about how we performed phenotyping in this experiment because Heino already previously talked about it, but if you have more questions, please ask afterwards. And yeah, so basically we applied mixed linear models, for example, in the software Tassel, as well as latent factor mixed models to account for genetic structure as modeled as latent factors. And we use different models of different complexity by also including the effects of genetic structure, kinship, as well as other environmental variables such as altitude and the year of termination. And similar to what Giorgio showed before, we also were not very successful at identifying SNPs that were associated with the resistance. And actually also found in the best model two SNPs that lay basically on the same chromosome and on the same contig from the older assembly, and that are found in a amidrohydrolase gene. So afterwards, I thought, okay, previous works already showed that this is actually a quantitative trait with a highly polygenic basis. So I thought maybe, despite the fact that we are not able to find SNPs that have a very strong signal after FDR correction, still maybe it's possible to identify a part of the data which is showing some trend between or softer shifts in allele frequency between resistant and non-resistant individuals. So I applied a multivariate approach, which is called conditioned redundancy analysis, to try to find such loci. And so this multivariate approach indeed managed to a certain extent to find a small part of the data, which is explained by resistance. So basically by this third redundancy axis in this plot, which separates basically the resistant individuals in blue from the yellow one, which are susceptible. And a bunch of SNPs here shown in pink, dark pink, and as triangles seem to be quite separating quite well these two types of individuals, these two classes. And these are actually SNPs that show such a pattern. So if you look at this plot here on the left, you will have in each, for each genotype, you would have both sick and healthy individuals, but you still have a higher proportion of healthy individuals for one homozygote genotype, such as for this SNP, while you have a higher proportion of sick individuals for the opposite, the other homozygote genotype. And actually I thought, okay, maybe this is actually really interesting loci to a certain extent, because I also saw that some of them, a few of them overlapped with previously identified candidates from Stock et al. 2019, and 10 of them were in closer proximity, so below 5 kb distance from previous candidates. But then I unfortunately saw that many of these candidate SNPs were actually driven by rare genotypes, so not rare alleles, which were already filtered out previous to analysis, but rare genotypes, such as in this case, where you see that one homozygote, the AA homozygote is actually represented just by, or carried just by 6 individuals in our data set. So of course, there is not very much statistical power to establish if this is a true candidate or not. So with these, I would like to bring some concluding remarks about our analysis so far. So I would say that both mixed linear models and multivariate approach actually failed to detect strong association between these SNPs in the array and the resistance. Importantly, also, most of previously detected candidate SNPs are apparently monomorphic or carry just rare variants in Austria. So for now, we were not able to establish if the 4-tree array does really deliver decisive information on resistance in Austria. And yeah, with this, I would like to thank many people involved in the work, and thank you for your attention.
Thank you, Aglaia. That's really interesting. And so finally, on people who've used the 4-tree array, Miguel Nemesio will speak about use on Irish materials and also some Dutch materials as well.
Yeah, can you hear me well?
While Miguel is loading up, I'll just say that these were the only projects I'm aware of using the array. If I missed out any, I do apologize for that. So any omissions are inadvertent on my part. Miguel, over to you.
Hi, Richard. Can you hear me well? Yes. All right. Yeah. So I wanted to introduce. I haven't worked on ASH for a while now, but I basically will present the last bit of work we did on ASH before I finish my role in Ireland. And basically, this is all information coming from Will's chapter 6 of his thesis. So what we did was to sequence 3 different small groups of plants. And well, here I introduced in total it was almost 500 genotypes, and they are divided in 3 groups. One would be a population, which is basically the TGB or the Chagask gene bank. So this is a collection of over 200 putatively tolerant genotypes coming from all of Europe. So it has a very broad background. Then we have the MET population, which is the same genotypes that were used for my metabolomics paper. So basically here we wanted to see if we could identify signals, genetic signals associated with specific metabolites, which are important for defense against dieback. And finally, the HK population, which comes from our collaboration with IMBO, Marek Stenaker's group. And these were 2 half-seed families from 2 tolerant trees that were very well characterized. And yeah, about 100 half-siblings per family. And then five traits were measured in these trees, and we trust that these measurements were quite accurate. So I'll start with the first one. The Irish collection of tolerant genotypes, it's a collection created between 2017 and 2021. And nowadays it's established as a gene bank or a seed orchard in 3 different locations in Ireland. And well, this originally included 218 putatively tolerant ASH genotypes with multiple European origins from areas which were supposed to be compatible with Irish climate. 39. So most of these genotypes, we kind of got them from different trials in Europe, especially RAP trials where tolerant individuals had been identified. And then some of them were actually part of almost 1,000 Irish genotypes that were sent to Lithuania for 4 years. So the thing is that in Ireland, the program started before the disease was heavy enough to select trees in Ireland. So we either had to bring them from abroad or we had to actually send a lot of Irish genotypes somewhere where the disease was very bad. So that's what we did in Lithuania. And after 4 years, we got 39 genotypes out of 945 that we sent, which showed a moderate or high tolerance. And then, yeah, we found different levels of testing because they were coming from different backgrounds. So we saw that some of the trees, once they were planted outside in Ireland and the disease was a bit heavier, they showed that they were susceptible and intolerant. So they hadn't had enough rounds of testing before being selected. And well, here what we did was to use 10,381 of the SNP markers of the array. And here. And well, what we saw first was that we could more or less divide the trees by provenance. So depending on what country they came from, they had like genetic imprints from that area. Surprisingly, the Welsh ones were very different, but well, that's a curiosity, I guess. And well, what we did too was that to see which of these trees actually had, here in gray, in dark gray, you can see the trees that actually showed different proportions of being susceptible based on how many ramets of each genotype had damage. And we were trying to see if we could use the genomic prediction on these trees that had fallen through the way. And well, it was not possible for this population. And what we thought is, well, you know, as long as this is the genotype for the trees that are in the breeding program, in the Irish breeding program, we have that information and they will be monitored in the future. So hopefully with more data, when we have better genotyping and in a more homogeneous environment, we can somehow recycle this data and see if we can get something out of it. Then again, the second set of trees was these metabolomics trees, which were used to identify metabolite markers. And here we have 20 highly susceptible and 20 highly tolerant fraxinose successor genotypes with very different backgrounds as well. And in terms of where they came from, we identified 64 candidate metabolites. 39 of them were associated with lower susceptibility to ASH dieback. And well, what we did was to use, we had classified these trees based on if they are tolerant or susceptible. We felt that was a bit lacking. So what we did in this study was that we could use individual signals of metabolites and reclassify the trees based on high or low abundance of these metabolite in each of the genotypes, and reanalyze this and see if we could find SNPs associated with a higher or lower presence of these metabolites. And well, we first, again, we saw this, again, this geographic component to it in the preliminary test. And then later on, we found like actually different markers associated with different metabolites. And well, in a way, this is, the intention of this is to divide the mixed signal of this multigenic trait. If we can identify, chemicals which we can actually measure individually, and we can associate SNPs to these metabolites, then we might be closer to finding some markers that mean something, or mean something at least that we know exactly what it is. And well, this is a bit the results of our first kind of round of analysis there. And here is a list of the different markers that were associated. And again, we have like what kind of protein they are encoding. And in most cases, or many cases, they would encode enzymes which are directly related to these metabolites. They all have quite a high significance, and I guess this is a good database to work on. And finally, we have this offspring of two tolerant genotypes shared by Imbo and Chagas as part of their collaboration. And of course, Richard's group was also very involved with this. And well, I say putative half-SIP families because in fact, many of the half-SIPs were indeed full SIPs. So we could assess that later. And well, the thing is that we had two independent families that we could use to study different traits. And these families had been very much monitored at Imbo, and they are still producing data to this day. So we had the chance to have several rounds of testing artificially and naturally, inoculation. And well, again, like here we can see clearly the difference between the two different families. And then again, like we found different traits: bud burst, height, diameter, and then artificial inoculation or natural infection of leaves. And again, we found several loci. And there was again a reasonable overlap with markers already reported by Stokes et al. In 2019. So we found that, a part of the signals are shared, and perhaps we should focus on those that are shared and dig further there. And finally, we tried to do prediction with all these groups, but it wasn't possible. We didn't find any strong signals in terms of prediction for the different groups that we looked at. And well, yeah, as I said, like this is work that was carried out by Chagas, and of course this includes a lot of different groups in Europe. I would like to highlight the help from Richard and his group, and of course IMBO as well, which was a main provider of tolerant material for us. And that's it.
Thank you very much, Miguel.
Mm-hmm.
Now, the rest of the meeting is going to be two discussions. One looking, in some ways, looking backwards and thinking how to publish the data that's already been generated. And Arnaud will lead that discussion. And then there'll be a discussion about what do people do in the future to move forward for future genotyping, especially if they want to be predicting ASH dieback tolerance. And in between those two discussions, there'll be a short presentation from Joanna Meger from Poland, who's been doing some genotyping with whole genome sequencing. So I'll hand over to Arnaud now to lead the first discussion. Thanks, Arnaud.
Thank you. Do you see me? Do you hear me?
Yes.
Okay, good. I already told you my point of view in my message before Richard organized this meeting. Many of you, if not all of you, reached the conclusion that the bee forest or the 4-tree array doesn't lead to any result in terms of genomic prediction or GWAS for ASH dieback resistance. But we do not have better results than you, but maybe a slightly different point of view. In Austria, so Aglaia, you said that heritability of resistance to Caldera is quite high, and you give values of 0.3 to 0.65. First question, is it narrow or broad sense heritability that you computed? Did you use the array to compute the heritability with the real genetic distances? Okay.
Maybe this is a question that Heino can answer better than me, because it was analysis from Professor Kirizits, I believe.
Okay.
Heino, you're in the audience. I think you can answer.
Yes. Well, actually, I have to give the question on to Gregor Unger. He has been doing the experiment.
Okay, I'm saying that because I think that there are two publics in this audience. Some of us are breeders, and some of us are molecular geneticists. And as a breeder, I do not consider heritability from 0.3 to 0.65 as a high heritability. We computed and published the heritability of tolerance to ASH dieback, both at the crown level and at the collar necrosis level in 2016. And we reached a value of 0.4 for a narrow sense heritability. Giorgio will tell me if I am correct, but I think he computed values based on the SNP data, on the 4-tree data, and the value is now much closer to 0.2 than 0.4. Which means that most of you molecular geneticists, you rely on phenotypic data that is provided by breeders. But I would like to ask you, have you ever met a breeder who provided you with an ASH genotype that has proven to be tolerant or resistant whatsoever, and that has been tolerant forever until now? Because to my knowledge, none of us has selected any tolerant, durably tolerant genotype until now. All of us know that when Caldera is progressing in a stand, in the end, you got maybe 1 or 2 trees that remain free of any symptom. And then when you start grafting them and put them in another trial, that's what the people from Lithuania did a long time before any of us. They thought they had selected tolerant genotypes, put them in a clonal trial, and in the end, none of the clones remained free from the disease. So what I want to say is that, okay, there might be a problem with the array. I don't know, because I'm this time I'm not a molecular geneticist, so I'm not good in analyzing this. But please be conscious also that there is a big problem in the phenotype. And when I see people saying that they've got 20 or 30 or even 50 tolerant ash trees, that's just impossible. I do not believe you when you say these numbers. So that's something that I told you already and that I wanted to point out again. The heritability is not high at all, and phenotypes are not reliable at all. Now, putting this aside, we all see that when we are using the array, we're seeing a clustering of populations, and I want to share some hypotheses and some ideas with you about what we might discover or rediscover using this data. I will share some slides with you. Okay. Now. Do you see my screen?
Yes.
Yeah, good. So first thing that has already been illustrated by Giorgio and some of you is the fact that using the array, we can discriminate between Angustifolia and Excelsior. Because as I said before, we do not know what we are talking about when we are talking about tolerant genotypes. But do we really know what we're talking about when we're talking about Excelsior genotypes? When you start looking for potential hybrids, because we know there are spontaneous hybrids between Excelsior and Angustifolia, when you start looking for them, in the end, you find them. And so this has been shown already by Giorgio. Sorry, this one. So you all know the natural area of Excelsior and Angustifolia. You know that there is some overlap between the natural areas, and especially along some rivers in France, but also in other places in Europe. There's been several publications already on the matter. In this publication from 2013, you see that you can have hybrids up north. Sorry, to me English is a major problem. You can find hybrids, or you can expect to find hybrids in Germany and even in Lithuania when you are along some river basins. In orange here you got Angustifolia genomic how to say that, print, genomic pool, and in blue the Excelsior pool, and you can see some blue dots north in Europe, where you got a line indicating that you can find a few hybrids or a few some Angustifolia genomic pool, genomic content. And there's been some studies on the matter in France, things that have been published at the beginning of the century, and along two main rivers, which is the Loire Valley and the Saône River, and in these two environments, hybrids were found using only 5 microsatellites. And I want to show you also something that maybe you did not notice before, but which I will relate to what has been presented before, which is that when they use these 5 microsatellites to try to find the hybrids, and they run a structure, and using a number of a k number of 3 in the end, which is the best number they found. In the end, they find 3 ancestral populations in all these populations that they studied in France. So in black, you got the Angustifolia genetic background, but for Excelsior, you can see that they've been able to discriminate 2 ancestral genetic backgrounds in white and in gray. And this made me think about the fact that in Fraxinus excelsior, many scientists before us, botanists, defined ecotypes. And especially in Germany, in Austria, in Romania, I think, there is a lot of literature on that, on the fact that there would be at least 2 ecotypes of Fraxinus excelsior, one from dry calcareous sites or mountains, and one from flood plains. And my question is anybody of you familiar with these ecotypes in your country? Can we get material from both ecotypes? Do you know the people who conducted these studies? Because when I see the results from Austria, and when I look at the geography of Austria, altitudes, I tend to think that maybe the 2 populations that you find with the 4-3 array are just a signature of these 2 ecotypes. And I would find it very interesting to merge our data sets, at least with the altitude data of the trees that were sampled, of their environment, their original environments, and see if we can confirm my hypothesis that we are discriminating these ecotypes, because these ecotypes have been hypothesized a long time ago. See, I've not been able to find this publication. If any of you can access it, I would be very interested. It's from 1925, and certainly in German. And if these ecotypes have been hypothesized, no one has been able to demonstrate any genomic signature of these ecotypes. So I'd be interested in merging our data sets to see if we can sort things out, and maybe we will discover that when we're talking about Fraxinus excelsior, we are talking about something more complex than we think. Also, I wanted to point out that in the classification of Wallander that I think we are all using when referring to Fraxinus, the only 2 species for which Wallander defined subspecies are Fraxinus chinensis and Fraxinus angustifolia. And we know that the array is able to discriminate between Angustifolia and Excelsior, but I would be very interested also in seeing if it's able to discriminate between the subspecies of Angustifolia. So my question is, are you familiar with these subspecies? Most of you are from northern countries, so you're not much concerned with Angustifolia. but I'd be interested in including data on angustifolias from Southern Europe and see if we can discriminate between the subspecies. Because maybe these subspecies have different behaviors also in terms of tolerance to Caldera. Maybe some of these subspecies are closer to Asian species than the others, and I'd be very interested in doing this. But I'm not sure this is the audience I should or I could spend. How can I say? I'm not sure we got the data at the moment or the genotypes at the moment to study this this matter. Okay, so that's all I wanted to say. So when we look at the results that we all got from the 4-tree array, we see that we got groupings of populations. I tend to see something that has something to do with altitude or sorry, with latitude. Okay, so I think the more data we can gather, the more clearer it'll be. sorry for my English. The discussion is just open.
Yeah, shall I? Because you talked about the excelsia from let's say the normal sites, the wet sites, then the sites in the mountains with the calcareous soil, the substrate. The pattern we see in Austria, I think, is not showing this, but it's showing rather. I think the situation is even more complicated than that. So because we do not find differences in the excelsior, whether it's from the mountains or from the lowlands. This pattern we found is purely east-west. So in the west of Austria, there's a strong. In the west, there's a strong different cluster. We have found this also in other tree species.
I do not know much about geography, but I thought altitude was higher in the western part of Austria than in the east.
But it's yes, that's right. But actually, in that part where the ash was collected, is not so high up, let's say. Okay. It's also not on calcareous soil. So we have many populations also in the higher mountains, but they don't show genetic difference to the ones to the other ones. So I think that's more a topic of the post-glacial recolonization and not associated to the site. But yeah, of course, you could look deeper into this matter. Actually, that was also our plan to analyze our data more in that direction, also in the future, in the coming weeks. For the angustifolia, I cannot say much. We have few angustifolia in the east of Austria. They are all oxycapa, and but surely I can imagine that there's a difference between east and west of Europe, also in that species, quite strong.
Okay, thanks Arnaud and Heino. I think I'll have to invite Joanna to give her talk now. Perhaps the discussion of whether or not all of these population studies are published together or separately can be pursued another time, perhaps by email. But let's hear what's been going on in Poland. Thanks, Joanna.
Thank you, Richard, for the opportunity to talk about our project genetic research as a rescue for Polish ash trees.
Okay. Our team has developed a new genome of Fraxinus excelsior. This reference genome was generated from a female tree with potentially resistance to Heminosypus fraxineus from the botanical garden at our university. The assembly utilized Illumina, PacBio, and HiC data. This yielded a high-quality chromosome scale reference genome with 23 chromosomes and an additional 4,415 scaffolds. The total length of the genome was 806 megabase pair. Of the 2,326 Fusco groups searched, almost 98% were complete and present at least one in the genome. Our genome is available in the CBI database as genome assembly frax 001 PL. Using known plant repetitive element sequences, 59% of the genome was identified as repetitive. Gene annotation yielded a set of almost 41,000 gene models. We are still performing additional analysis and preparing the manuscript for publication. Last year, we received funding from the Ministry of Education and Science for the project Genetic Research as a Rescue for Polish Ash Trees, headed by Professor Jarosław Burczyk. As part of this project, we collaborate with the Forest Gene Bank in Kostrzyca, which has gathered information from forests about the occurrence of resistant ash trees. Last year, we collected material from over 100 reported ash populations from all over the country. For each population, we selected 3 trees, 2 healthy and 1 diseased. Within these trees, we selected at least 1 female and 1 male. As a result, we have 337 trees, including 223 healthy and 114 diseased trees. DNA isolation is coming to end, and soon we will send our material for whole genome sequencing. In the picture, you can see a typical example where tree is dead, likely due to Heminosypus fraxineus, next to potentially healthy one. The conditions of trees was assessed based on synthetic tree image. This index was calculated for each tree and may take values from 0 to 3. An increase in the index value indicates a decrease in tree vitality, whereas reduction shows its improvement. Individual values of synthetic index corresponded to respective degrees of tree damage. 0 was healthy tree, 1 weak tree, 2 damaged tree, and 3 dying tree. We collected also several additional metrics such as height, diameter, and health of trunk. Our main goal of the project is to find the genes and markers responsible for resistance to Heminosypus fraxineus. We would like to develop a set of markers that will be efficiently enable the selection of resistant trees and will be used to create a breeding population for the production of forest reproductive material with increased resistance. Our second goal was to identify the genes related to sex determination in ostriches. We will probably have funds for additional sequencing of samples from Europe to test our set of our markers on a larger scale. We're currently finalizing the details. If any of you are interested in cooperation, I encourage you to contact with us. Thank you.
Thank you very much, Joanna. So now I'll hand over to Heino to lead a discussion on the future. What should we be doing as a community in future to try to move things forward for the benefit of breeding programs? Thank you.
Thank you, Richard. Yeah, I think we can immediately jump into the questions. René has the hand up.
Yes, thank you. Well, 2 points. First of all, if Arnaud concludes that the array is not useful to find resistance, and then concludes that resistance in fact doesn't exist, I think either of the conclusions can be correct, but not both, because if trees that are healthy are only healthy because they happen to be not yet infected, if you come back a few years later, they have been affected, and it's purely a geographical effect and not a genetic effect, that would then suggest that you don't find a genetic signal for resistance because it doesn't exist. So then the array would not be good or bad, but it just it's indifferent because what we have seen as results is possibly artifacts or happens to be valid in some populations but not in others. Perhaps it's good to add, because I realize I forgot that in my presentation, we did not select and throw away SNPs from the selection that was made in the UK, other than on technical grounds. So all the SNPs are there. So apparently all the SNPs that are gone that gave a good signal in Carey's study, they probably had a poor performance in terms of separation or minor allele frequency or those kind of things in our hands.
That would make sense, because we didn't sort of filter for particularly minor allele particularly strictly in the pool. It was so yeah, likely some of the SNPs that are included there are not good ones for inclusion on an array. I think that's fair to say.
Maybe coming back to the question if there are resistance markers or not, I think what we have discussed during the webinar series, what Richard I think has said, there's of course there's a certain degree of resistance in the ash, but if there is very high disease incidence, this resistance is strongly tested, let's say, in the wild. So it's I think that's a problem, because it's of course polygenic trait, and to find the resistant trees in a high disease incidence situation, and there's so the heritability is probably not so high, and therefore it may be the environment has a very high effect, of course, on this. So that's, I think, what Arnaud has also pointed in that direction. Yeah, what I take from the discussion today is that the SNP array, as we have found it also in Austria, is maybe not sufficient to detect the genes in question, or the markers in question, markers we need. So that it is important to have more genomes, and to develop the methods further. Maybe Joanna, you or Arnaud, okay, you have the hand up.
I forgot to tell you that since we reached the same conclusion that we could not detect anything using the final array, when I got the news that I had some money to spend to add some more trees genotyped using the array, I decided not to use the final array, but to go back to the pre-screening array to get much more SNPs. But the genotyping has been done, but we haven't analyzed the data yet.
Okay, that is also done by Thermo Fisher, or?
Yes.
Okay, well, that's a good point for practically, yeah. Barbara?
Maybe I have a question to that, because we are just now planning to use the array as the final array, and just about to send the samples to Thermo Fisher. So this would be kind of an option for us also to use not the final set, but the preset. So that would be maybe we could discuss or to see how we could also use maybe the full set.
Of course, that means that you will genotype less trees for the same amount of money. I cannot remember the difference in the cost difference. Maybe Veronique can tell us.
Yes, for the same money, you genotype 1,000 genotype with the final array, and we genotype 288 genotype with the pre-screening array.
So between 3 and 4 times more expensive.
Yeah. Because we were told that we have to send a minimum of about 800 samples only to, so 2 plates for to start even the experiment. Yeah.
No, you can send, yes, we sent 2.
Okay.
280.
Yeah.
3 plates.
So, yeah, that seems to be the available technology at the moment, besides the whole genome sequencing.
Yeah.
Another point is maybe the phenotyping. That's also what Arnaud has hinted on, that it has been, and also in I think in Giorgio's presentation, that the phenotyping, if you have only the defoliation, it is very complicated. So in Austria, we have been using the dieback on the woody parts of the offspring trees, but of course this you cannot easily do in adult trees. But what gives me, what should be done, I think, and that's these lesions at the base of the trees.
Yeah,
that's something that's also correlates well with the findings of that the lesion length. If you do artificial inoculations, this gives you also good indication of the resistance. So I think we should put more emphasis on proper phenotyping. Otherwise,
Yeah, I think if you use two integrative phenotypes, then you do not find anything. You need to focus on a component of the response of the tree. And I agree with you that we should pay more attention to collar necrosis, as they look like what Giorgio analyzed when he analyzed the lesion lengths after inoculation.
Right, that's yeah, Carey, go ahead.
I might have missed this earlier because of my connection, but when you're measuring the lesion lengths, is that scaled to the size of the tree, or are all these trees reasonably young, sort of field trial trees, as opposed to like mature woodland trees?
Are you talking about lesion lengths after inoculation or basal
So
necrosis?
is this all sort of deliberate inoculation when you're measuring lesion lengths? So that's only looking at trees where you've, okay, that makes more sense. I missed that bit.
Yeah, so that's in young trees, I guess that was done in the nursery. Yeah. Okay.
Could I
Yeah,
just
Richard?
make a few comments that strike me in this discussion? Is one thing that's really interesting is that some of the loci that have previously been associated with resistance, the minor allele frequency is very low, which makes life difficult, but in a way you might expect that, given that so few trees are showing any resistance anyway. It's not surprising that they're low frequency alleles.
Exactly.
Broadly, I agree with Arnaud's point that there are no trees out there with complete resistance, but for this genetic work, you just need a relative difference. You need some trees that are showing more resistance than others. And as long as you can divide into two categories, then it's okay for this genetic work. So I don't think it's all meaningless just because none of our trees that we're basing things on are fully resistant. If some of them were fully resistant, we wouldn't need to do this work anyway, because we could just be selecting those fully resistant trees. I think perhaps we're being too negative, though, about the 4-tree array. I'm leaving this conversation thinking that it's actually somewhat better than I feared. There have been some associations shown, for example, with the lesion lengths, which are probably a much more reliable phenotype. So that's encouraging. So I'm not sure we should be giving up on the array. And I wonder if one of its uses, although it can't give, a really certain prediction, I think if someone is doing a breeding program and they're choosing trees to cross, it would still be worth getting a genotype from the 4-tree array, just to make sure that you're maintaining genetic diversity and you're not just crossing two very closely related individuals. If you're choosing among a large number of individuals and all show a similar level of resistance, you probably want to pick two that are genetically different. And you could use the 4-tree array to help you with that. And as a side benefit, it might give you some hints about resistance, even though it's not giving you absolutely certain predictions. So I think there is still usefulness for the 4-tree array. And then the final comment I'd make is a much more practical one, and that is, when will the 4-tree array paper come out, so that other people can be publishing their results? Because I think that might be a bit of a limiting factor in people publishing.
Yeah.
Perhaps I can say, because I know Patricia left it for a while, so I claimed that I could help her. So now it's on my desk. So I try to limit the delay as much as possible.
Top of the desk.
Yeah.
Yeah, Jarek, you have the hand up.
Yes, I thank you that we have the opportunity to join this discussion and say a few words about our ongoing project, what Joanna just said. We have probably quite different perspective, and Poland was the country which was found to be one of the first with so heavy damage of ash due to cholera. So we wanted to do this work from the beginning. Indeed, phenotyping and saying who is resistant, who is not, is very difficult in the field. Among the old trees, most of those trees are 100 years old or older. But we saw some cases when side by side in the same environment, we had two individuals quite different in their response to the presence of the fungus. So we still believe that somehow from the early papers that there is some within population variation in resistance. So we chose the design. Joanna said about this, I just repeat. We selected the populations which were known to be infected. And in those populations, we were searching for most healthy individuals and sick individuals. We selected 2 healthy and 1 sick, and we'll go back in 3 years and we'll see how many of those healthy are still healthy. But I think that the idea was to make the whole genome sequencing of a large number of individuals. And we hope that the large number here is a point from the very wide area. So you notice we sampled just 3 individuals within the populations. So we have very broad genetic basis. In terms, there will be very, very low kinship in this material. And what I'd like to say, and Joanna said this clearly, that we, in our project, we still have some room for whole genome sequencing of additional individuals. We're just counting money. And I think this might be something like 100 individuals extra. So I encourage you to contact with us that if you have some beloved healthy trees and sick trees, and you didn't have your funds now for whole genome sequencing, I just contact with us, because maybe it's a good idea to have sampling across Europe with sometimes 2 or 3 populations in every country who might have good examples of side-by-side growing healthy and unhealthy trees. And spring is coming, and I think this is a good time for sampling. And what we learned from our previous study, we have some initial sequencing of about 30 individuals, and the genome size is very variable, from something like 750 to 900 megabases. So there is some room for among individual variation in genome size. We noticed that most of the variation is caused by repetitive elements. But I think that pangenome of the Fraxinus is not well understood. And I think that the idea of working on the pangenome is always better when you have large, broad samples across the distribution of the species, not just from a single country. And I think, of course, all the data that we will generate after sequencing will be available back to you. So you will have your access. You don't pay for this. We are just interested to have the background, which will be wider than just a single country. So if you are interested, just contact to Joanna, and we are just ready to deploy our data. And anything else, if you, by the way, can identify male or female, the sex of your individuals, it could be even better. For details, just go to Joanna. So I'm very happy to be with this audience, because this is the audience of experts who know what's going on with ASH and fully understand the idea of having everything sequenced indeed. So I think we still have to do this work. We're not promising it will be successful, because we know that partly depends on the quality of phenotyping, which is genotyping becomes simpler than phenotyping. So I think, but this will take some more time. But we'll be very happy if you consider that maybe you have some individuals that you want to be sequenced. We're using just Illumina, but it's quite useful for many cases. Okay, thank you.
Okay, thanks, Jarek. Thanks. Good point. I think Laura was first.
Oh yeah, thank you. Yeah, so well, I had a question about the lesion lengths, but I would just also mention that so I think some of you are already aware, but we have a project that started in the last year, funded by DEFRA, to start to build a pangenome for Fraxinus excelsior. So that's using long read data, building on an improved linear reference, which is for the same individual that we published the original reference genome for in Nature in 2016 or 17. And it's biased more towards UK provenances, but we are also including data from trees that originated from other countries throughout Europe. And I'll share I'll share a link to the project page for that in the chat in case that's of interest. And Danny Wood, who's the postdoc, who's basically doing all the actual work to build the pangenome, is on this call as well. But no, in terms of the lesion lengths, I just wanted to ask, so I think, you know, some years back, there was kind of discussion about the fact that we perhaps thought that these kind of phenotype scores from artificial inoculations might be less representative of sort of actual resistance that we might see in nature, because it already kind of subverts some of the resistance mechanisms because it's involving actual damage to the tree. So it's interesting to hear that you say that it seems like they are quite representative of, or at least correlate well with the basal necroses you see. So I just, yeah, was wondering if it is now thought that perhaps those are a good measure of the level of resistance we might expect to see in nature. And I don't know who's the best person to answer that question, or if anybody has any general thoughts on it.
Not sure, maybe somebody wants to answer.
I
Giorgio?
don't know, maybe I can say a few words. To me, so I'm not an expert on ASH, first of all, so that's important to know. But to me, this lesion length is a subtrait. So the point is that perhaps measuring better a subcomponent of a more complex phenomenon. So that's the point. I don't think we can say that. It's just something more measurable, perhaps.
Yeah, I could even generalize that. If I look around in our department, we do a lot of disease resistance, research on disease resistances. And if you move to a new crop or a new disease, the first hurdle is a proper phenotyping test that is sufficiently sensitive and quantitative to really get good results. And based on that, you can do then the genotyping. As was already said here, phenotyping becomes more crucial than genotyping. But I think it is important to tease apart this kind of complex multi-gene resistances into some of the components, because if it's really a lot of genes and aspects that are involved, you may not easily get trees that are really sufficient trees that represent combinations of genes in order to tease them out from wild populations. And if you zoom in on the components of the disease resistance, you may be able to, as Giorgio basically already showed, you may be able to find genes or loci that correspond to that component of the disease. Now, of course, how representative that component is for what you actually see in the wild, that is the second question, because you don't know all the components. And sometimes they can be useful in rather different ways. A colleague of us, Erik van der Weg, studies apple cancer, which is also a fungal disease. And he has actually two completely different experiments. One is on the incidence of the infection, where he has natural infection in some kind of assisted way. And he also is interested in how fast the lesions then grow once the tree is infected. And for that, he has a completely different setup in which he makes artificial infections, because otherwise he doesn't have enough infections to measure it. And different trees come out. So those are different components of the resistance that you find in different genotypes. But you can measure that the outgrowth rather efficiently and precisely. And the use of the outgrowth is that if the tree is slower, if the outgrowth is slower, it gives the manager of the orchard more time to actually go in and remove those trees. So it doesn't help you to avoid the disease, but it does help you to manage it in the population. So some components can be useful in slightly different way, perhaps than you anticipate beforehand. So I would really think that if you think about better phenotyping, think about what components would we actually be able to measure, in the hope that will, the results of those tests will be help you to find actually the genetic components underlying them.
Okay, thanks. So now Arnaud, you can go ahead. Yeah, it was 1 millisecond.
Yeah. Okay, that's exactly my point. That we are taking two integrative phenotypes, and that we should try to use the genomic data on components of, I prefer to say susceptibility. I take it the reverse instead of saying tolerance or resistance. And that makes me think also that I wanted to propose you something, which is certainly many of us got other phenotypes on the genotyped trees than resistance to Caldera. As an example, I got a lot of phenological data about bud burst, and I would find it very interesting to make the same kind of analysis on a good heritable traits, easy to measure, reliable, like phenology. So if you've got phenological data, maybe we could try to run the same analysis and compare our results at least. And I wanted to say also, because Jaroslav talked about sex in ash, that I got the feeling that sex in ash is still underknown. I don't know if that's correct for many of us, because many of us stick to the idea that sometimes that hash can change sex from 1 year to the other, which is wrong. They can marginally change, but a male never becomes a female. So there are males, females, there are hermaphrodites also, hermaphrodites with female tendency or hermaphrodites with male tendency. And Jaroslav, as you are asking for males and females, and Richard also, as you said, we should try not to cross genotypes before checking that they are sufficiently genetically different. In the discussion, I attached a paper that we published in 2018, where I want to draw your attention to the fact that there are also something that is an incompatibility system in ash. So there are two groups in common ash, two groups of compatibility. And obviously, many people did not notice this publication, and I would find it very interesting also because this is a project also that we have with a university here in France, to make a whole genome sequencing of ash trees belonging to these two groups of compatibility, and to try to find the genes responsible for this incompatibility system. So this is something that I will discuss with you by email, certainly, Jaroslav. But I would like all of you to be aware that the sex in ash is very complex, more stable than what people think, but more complex. And last thing, yes, as a breeder, of course, I prefer to think that there is resistance in ash, and that in the end we will be able to select resistant or tolerant ash trees. The problem is just the reality that I am facing. I have been measuring more than 10,000 trees. I selected 60 trees among 10,000, grafted them, put them in a clonal trial, and 2 years later, none of the genotype, 0 of these genotypes were free from Caldera on all their ramets. Okay, so that's disappointing.
Maybe I can add on that also a little bit, but Jarek, you also have a
yeah, just a very short comment or rather question. When we did some preliminary analysis 2 years ago with double digest rod sequencing of ASH, we tried to see how many those rats mapped to Caldera genome, and actually it was we didn't finish these analyses to the end, but it looked like there were rats mapping to Caldera genome both on healthy and unhealthy trees. We didn't quantify this, but I think that now with the whole genome sequencing of leaves, and we sampled our leaves in June and July, so there's the timing when the healthy trees could also be infected or be present. So I'm just asking, have you heard about the presence of the cholera or hemanosyphilis, whatever it's called, in the tissue of healthy trees? Because this is common to René suggestions or comments that this is the problem, how the tree responds to the presence of the fungus, not if it is just sensitive or insensitive. So I think that in many cases there is a pathogen, but the individual tree responses at this particular moment in a specific way, and I think it's very difficult to find genes that correlate with the response of a tree in particular environment, in particular ontogenic situation. So if you have this comment or experience that in the genome in individual which looks healthy, there's still cholera fungus present, it would be probably possible. But I think that with the whole genome sequencing, we can quantify number of DNA, amount of DNA or number of rats that map exclusively to unique genomic regions of hemanosyphilis. So I think that this might be important for the future. Thank you.
Yeah, it's an endo fight, and yes, it's not surprising when you find the fungus also in healthy ones. But Eric, you have maybe you have more experience.
Yeah, just along these lines, I mean, our experience is that we do find the fungus also in the I call them tolerant, exactly because we find the fungus in the trees that are not showing any symptoms. Our experience is actually, and I agree with Arnaud that we shouldn't be surprised if you go into natural forest and select healthy and unhealthy trees and cannot really find high heritability. But in the test sites, I think we can do that. And again, we're not, I mean, we shouldn't expect to get more heritability than 0.5 or something, but that's also very high actually for narrow sense heritability in the field trial. And when we have been phenotyping now the trees in the trial since the first measurements in 2008, so we actually have a long array of data, and we can see that some trees actually maintain their tolerance. We can also see when we do controlled process that, you know, that healthy parents get, or tolerant parents get tolerant offspring. And so I think there's a lot of I mean, I think there's a lot of evidence from the phenotyping that you do have some kind of high difference in tolerance so that you can build on. But the phenotyping does take time. And when we look at the data since 2008 and onwards, then, you know, I mean, there was a lot of trees that looked healthy for some times that then was not healthy. But there are also trees that maintain the high level tolerance. And when we look at the old trees compared to the young trees, we see that this signal, I mean, this expression take much longer time on old trees than in young trees. We discussed that a bit in this protein conference. Just to say that, I mean, so phenotyping, I mean, also we had some patients and it does take time. but definitely we do find the fungus in the trees. And also when we look at the trees we have now that have been, we think have been tolerant for a long time, we always see infected twigs, and so that's clear signs of infections. But the point is that the trees remain healthy. I mean, you see infections, but it doesn't really matter. It doesn't really spread into the stem. So that's just like, so I just want to add some optimism. And then I also, along what Richard said, that I mean, I actually personally I never thought, that I mean, what we really need is some kind of genomic markers that can really identify the healthy trees, because we need to test them anyway. We need to test other traits, as Arnaud said. We have this correlation between phenology and tolerance, and we are also, there could be other kinds of things that are correlated. So we really have to test the things in real life in order to do selection. They have to be tolerant, but also they have to do other things. So I think but the chip here would definitely be important for, as we discussed about diversity level, and also simply for breeding. As breeders, even though they're half-stitch, they're not really, they have the same branch history. So actually, so just for doing more precise selection in the breeding programs is really valuable to be able to estimate the coefficients of making this. But anyway, just to say that I feel confident that actually we have some trees out there that have a very high level of tolerance. Of course, it's interesting is how the fungal will respond to the fact that we try to introduce all these tolerant trees into the wild. But also there, I think we have a good case, because the biology of the fungus is probably not in the way that it will actually trigger a lot of selection for making the fungus more hygienic. Just some thoughts.
Thanks, Eric. Very, very important to have the overview. Yes, I think any more thoughts? Yes, please.
Is that me? Yeah. Okay. Great.
Yeah.
So I was just going to add that I suppose if it seems like from some of Carey's data that a lot of the tolerance that is predicted is from quite rare alleles, then it might be that there are not very many resistant trees, just because if there's like a bunch of really rare alleles, the chances of one tree actually containing them all might be quite low. Whereas if you did actually breed them all together, you might end up with a really resistant tree. And if it differs across different European populations, then maybe there's like a big pool of sort of compatible resistance alleles that might in the same tree, and that might not actually really occur in nature very much at all.
Okay, thank you. Yeah, so shall we close the discussion? Maybe.
Well, thanks for leading that, Heino, and thank you everyone for participating. And do drop me an email straight away if you've said anything that you don't want to be on YouTube. Thank you so much. Good to see you all.
Yeah, thank you for organizing.
Thanks for organizing.
Thank you very much.
Thank you so much.
Thank you. Bye-bye.
Bye-bye.
Bye.
Bye.